Okay, ladies and gentlemen, you are all welcome to my YouTube channel. If this is the first time you are coming to my channel, please subscribe. So in today's class, we are going to look at the concept of gluconeogenesis. And of course, as you can see, this slide is not made by me, it's made by some set of the students for their presentations. So I'm going to make use of this slide and make the explanation or make the video. So now, let's look at the objectives that we want to achieve in this video. Number one, we want to understand the importance of gluconeogenesis as an important pathway for glucose production, the main reactions of gluconeogenesis, the rate-limiting enzymes of gluconeogenesis, and then the gluconeogenesis as an energy-consuming, that is, anabolic pathway. So what is gluconeogenesis? Gluconeogenesis, the word gluco means glucose, neo means new, and then genesis means formation. So it means that gluconeogenesis is the formation of new glucose. So gluconeogenesis is the formation of new glucose. So the gluconeogenesis pathway is shown as one of the essential pathways of energy metabolism. It is an energy consuming pathway. Of course, since it is anabolic, that is the synthesis pathway, it is an anabolic pathway. So therefore, it must consume energy. So it usually occurs mainly in the liver, kidney, and sometimes in the enterocytes. Enterocyte, that is small intestine, is also occur in the enterocyte. Enterocyte, that is in the liver. So in the small intestine. It also occurs in the enterocyte. So these are the three part of the human body where mainly gluconeogenesis takes place, but it mainly occurs in the liver. So the targets of gluconeogenesis is to resolve glucose from non-carbohydrate carbon substrate, that is gluconeogenic substrate. So when we said gluconeogenesis in a simpler way, it means that it's a synthesis of glucose from non-carbohydrate carbon source, that is gluconeogenesis. And of course, the gluconeogenesis is both taking place in the mitochondria and chloroplast. So it takes place both in the mitochondria and in the cytosol. So if the substrate is glycerol, so it only occurs in the cytosol. But apart from the glycerol, all other substrates that are needed for the gluconeogenesis, some of them are localized in the mitochondria. So only glycerol is the one that is localized in the cytosol. So that is why when you have the cytosol, so you can easily use the glycerol in the cytosols and generate the glucose through the process of gluconeogenesis. So now let's look at the example of the gluconeogenic substrate, that is the substrate that I use in the gluconeogenesis. So we have number one, we have what you call glycerol, and of course this glycerol is usually obtained from hydrolysis of triacyl glycerol to produce glycerol and free fatty acids. And then of course another substrate for the gluconeogenesis, we have lactate and pyruvate. So this lactate and pyruvate, they are examples of organic acid, that is lactic acid and pyruvic acid. And then the next substrate for the gluconeogenesis is glucogenic amino acid. So amino acid that can be converted into glucose through gluconeogenesis, they are called glucogenic amino acids. So all the amino acids that can be utilized or that can be converted into glucose in the process of gluconeogenesis, so they are considered as a glucogenic amino acids. And of course, apart from the glucogenic amino acid, we should know that it's not all the amino acids that can make glucose. So there are certain types. And those other certain types of amino acid that cannot generate glucose through the process of gluconeogenesis, they are called ketogenic amino acids. They are called ketogenic amino acid. Ketogenic, they are called ketogenic amino acid. Ketogenic amino acid. So these ketogenic amino acids, they are used in the synthesis of ketone bodies. So these are the three major substrates of gluconeogenesis. Number one, we have glycerol, we have lactate and pyruvate, and then we have glucogenic amino acids. So these are the substrates that are used as a starting or raw material that are needed for the gluconeogenesis to take place. So then the next thing is the gluconeogenesis pathway. So in glycolysis, there are 10 reactions from glucose to pyruvate. And of course, remember in glycolysis, this is the glycolysis, there are 10 reactions, and let's look at them. Number one, the first one is when glucose is converted into glucose 6-phosphate by an enzyme called hexokinase or glucokinase in the liver. Then the glucose 6-phosphate will be converted to fructose 6-phosphate using isomerase enzymes, and of course from fructose 6-phosphate to fructose 1,6-bisphosphate down to pyruvate. So if you count them, you see that there are 10 reactions. But in the case of gluconeogenesis, there are 11 reactions. So we should understand that in gluconeogenesis, there are 11 reactions, while in glycolysis, there are 10 reactions. So this is one of the major, these are like some kind of the basic differences between gluconeogenesis and glycolysis. So glycolysis is only taking place in the cytosol, while gluconeogenesis can take place both in mitochondria and cytosol, except in the case of glycerol. And of course, 7 of the reactions in gluconeogenesis are the same in gluconeogenesis. So in gluconeogenesis, there are 7 reactions that are exactly the same with that of the glycolysis, and all those 7 steps, they are reversible steps. They are reversible reactions. And these are some of the key enzymes that are found or that differ in glycolysis and gluconeogenesis. So like there are 3 irreversible reactions usually in the glycolysis, and these irreversible reactions includes phosphoproptokinase 1, and then we have glucokinase or hexokinase. So these are the three irreversible steps of glycolysis. So since they are irreversible, so it means that in the case of gluconeogenesis, these enzymes are now replaced with another set of enzymes. So these are the 4 enzymes that are replaced from that of the glycolysis. And the first enzyme is pyruvate carboxylase. Then there also phosphoenol pyruvate carboxykinase, and fructose 1,6-bisphosphatase, and then finally glucose 6-phosphatase. So these are the 4 enzymes that are found in the gluconeogenesis that are absent in glycolysis. So they are replaced with 3 irreversible enzymes in glycolysis. So how is this glycolysis or gluconeogenesis started? First, when you have a pyruvate as a substrate, the pyruvate can be converted to oxaloacetate. So when you have 2 molecules of pyruvate will be converted into 2 molecules of oxaloacetate using an enzyme called pyruvate carboxylase. So from the word carboxylase, it means that the enzyme is adding another carbon. So CO2, that is carbon dioxide, is added to pyruvate to generate oxaloacetate. So pyruvate is a 3-carbon compound, while oxaloacetate is a 4-carbon compound. And then the next thing is an enzyme called phosphoenol pyruvate carboxykinase. So it's an enzyme that added and rearrange oxaloacetate into phosphoenol pyruvate. So in this case, a phosphate group is also added. And of course, from phosphoenol pyruvate, it will be converted into two uh, phosphoglycerates using an enzyme called enolase. So if you can see that from the enolase phosphoglycerate mutase, phosphoglycerate kinase, glycer, and then also glyceraldehyde 3-phosphate dehydrogenase, all these steps, they are exactly as the same with that of the glycolysis. So it means that it's only this step 1 and this step 2 and this step 3, and then lastly this step 4. They are the different enzymes with that of the glycolysis. So from this step 1, this final step, and enzymes, or the 11 enzymes are utilized, and finally glucose molecules is generated. So the synthesis of the glucose from non-carbohydrate sources is called gluconeogenesis, and it involves 11 enzymes. And these are the enzymes. It range from pyruvate, carboxylase, pyruvate, phosphoenol pyruvate, carboxykinase, enolase, mutase, kinase, dehydrogenase, isomerase, aldolase, and then we have fructose 1-cis, bisphosphatase. Of course, when you say phosphatase, from the word phosphatase is an enzyme that remove phosphate group. So like phosphatase is like a reverse action of kinase. Kinase is usually added phosphate group, but phosphatase remove or cleaved phosphate group. So of course that is why here we have fructose 1-cis bisphosphate, then but it converted into fructose 6-phosphate. It means that a phosphate group is removed at carbon 1. And then the next one is isomerase, which is an enzyme that converts the fructose to glucose 6-phosphate, and then another enzyme called glucose 6-phosphatase also remove the phosphate group here, and then finally you have the glucose. So this is actually the reaction. These are the reaction. These are the fat. This is the gluconeogenesis pathways that involve 11 enzymes, and these are the series of the enzymes that are involved in the pathway. So the next thing is actually the gluconeogenic substrate. So we are going to look at the gluconeogenic substrate. So we are starting with the glycerol. We are starting with the glycerol. So the first thing is, how do we obtain glycerol? So glycerols are usually made from hydrolysis of triglycerides. So when triglycerides, triacyl glycerol, and TAG, they are the same thing. It means that when you hydrolyze triacyl glycerol, then of course glycerol and free fatty acid will be generated. Glycerol and free fatty acid will be generated, and of course in the TAG, that is triglyceride, usually when it hydrolyzes, 1 molecules of glycerol is generated. 1 molecules of glycerol is generated. 1 molecules of glycerol is generated, plus 3 molecules of fatty acids. So it means that when you hydrolyze triacyl glycerol, 1 molecule of glycerol is generated, and then 3 fatty acids are also generated. And then there is an enzyme called glycerol kinase, and is only found in the liver or kidneys, and it is the rate limiting enzymes. So these enzymes, which is the glycerol kinase, is an enzyme that actually involve in the conversion of the glycerol to glycerol 3-phosphate. So it's an enzyme that add phosphates to the glycerol, and of course here we see that 1 ATP is utilized to generate glycerol phosphate. So it's a kinase enzymes. It adds phosphate group, and then The glycerol, the glycerol 3-phosphate will be converted into glycerol, dihydroxyacetone phosphate using an enzyme called glycerol 3-phosphate dehydrogenase. So of course, remember in the glycolysis, when we have dihydroxyacetone phosphate, then it can easily be converted into glyceraldehyde 3-phosphate. So of course, this glyceraldehyde 3-phosphate generated from the series of the action of two enzymes, glycerol kinase and glycerol 3-phosphate dehydrogenase, it will now be converted into glyceraldehyde 3-phosphate dehydrogenase, which will be continue off to the level where glucose where the glucose will be generated. So this is what we call gluconeogenesis, starting with the glycerol as a substrate. So dihydroxyacetone phosphate can be used for glycolysis or gluconeogenesis. So this dihydroxyacetone phosphate that is generated depend on the body demand. If the body is in need of ATP, then it will now continue with the glycolysis. But if there is enough ATP in the body, then it will now be channeled to gluconeogenesis to synthesize glucose. So that is glycerol as a substrate. And then the next thing that we are going to look at is the glucogenic amino acid as a substrate. So the catabolisms of amino acid are either through Krebs cycle directly or Krebs cycles indirectly. So like now, for example, glutamate and glutamine are the amino acid that can easily generate alpha ketoglutarate. Methionine and valine can generate succinyl-CoA. Phenylalanine and tyrosine can generate fumarate. Then aspartate and asparagine can generate oxaloacetate. And of course, you can see that clearly all the amino acids that can generate fumarate, because fumarate can easily be converted to malate, and then the malate can be converted into oxaloacetate. So oxaloacetate, of course, is a substrate for gluconeogenesis. So this is how it happens. This is how it happens. We can see clearly also glutamates and glutamine will generate also alpha ketoglutarate, and then the alpha. So you see the major aims of all these amino acid is to generate the intermediate of of the citric acid cycle, and this intermediate of citric acid cycle will be subsequently be converted into oxaloacetate. And once you generate oxaloacetate, then the oxaloacetate will be converted to phosphoenol pyruvate using phosphoenol pyruvate carboxykinase enzymes. So then after this is generated, then of course alanine and glycine, they are also amino acids which can directly generate a pyruvate. And of course, if it is the pyruvate that generated, it means that you need the actions of those two enzymes in the mitochondria, pyruvate carboxylase and then phosphoenol pyruvate carboxykinase. So these are some of the amino acids that when they degraded, they produce intermediate of Krebs cycle substrate, which can subsequently be converted to oxaloacetate, and then the gluconeogenesis will continue. So arginine can be converted to glutamate, then the glutamate will be converted to generate alpha ketoglutarate, but does not convert directly into alpha ketoglutarate. So of course, all amino acids are glucogenic. That is those that make glycerol, except leucine and lysine. They are not. Okay, then so alanine. So when we have alanine and glycerol, so does not undergo Krebs cycle directly, so it has to be converted to pyruvate. Then it will be later enter the Krebs cycles to generate the oxaloacetate, and then finally the gluconeogenesis will be continue. So then the next, so this is actually how gluconeogenic amino acid is utilized in the gluconeogenesis. And then the next thing is the lactates also as a substrate for gluconeogenesis. So glucose in the liver, it travels through the blood to the muscles, from the liver through the blood to the muscles, and in the muscles, the glucose can be hydrolyzed through the process of glycolysis to generate lactate. And the lactate at the same time can be transported back from the muscles to the liver. And of course, in the liver, the lactate will be converted back to generate another molecules of glucose. So this is a continuous process. Glucose from the liver transported to the muscles, and then in the muscles, the glucose will be converted into lactate, and then the lactate transported back to liver, then another glucose will be generated. So this is how the cycle continues, and this cycle is called Cori cycle. Like glucose in the liver travels through the blood to the muscles where it's turned into lactate via via anaerobic glycolysis, and then the lactate will travel through the blood back into the liver where it's turned back into glucose. Then it can go back again to the muscles again or any tissue. So this cycle is called Cori cycle. And note that lactate is released into the blood by exercising skeletal muscles and by cells that lack mitochondria, such as red blood cells. So, of course, the lactate is usually released into the blood by exercising skeletal muscles and also by the cells that lack mitochondria. So, it usually happens both in the red blood cells and the skeletal muscles, and this is described as a Cori cycle. Then the next thing is now we are now going to look at how pyruvate is being converted into oxaloacetate. So in this case, an enzyme called pyruvate carboxylase, which require a coenzyme biotin. So it requires a coenzyme called biotin. So this pyruvate will be converted into oxaloacetate, which is a 4 carbon molecules with the utilize of ATP and then carbon dioxide. And of course, this carbon dioxide is usually coming from carbonic acid. So pyruvate carboxylase, which convert pyruvate to oxaloacetate, is activated by acetyl-CoA. So acetyl-CoA is a biomolecule that activate this enzyme. So the activator of these enzymes is acetyl-CoA. And this acetyl-CoA is produced during a fatty acid oxidation, and it's increase in fasting. So during fasting, of course, the acetyl-CoA will be generated in the mitochondria from the citric acid cycle. But where does the carboxylation of pyruvate occur? This carboxylation of the pyruvate to oxaloacetate, it actually taking place in the mitochondria of the liver or kidney. But why? Because the enzyme pyruvate carboxylase is only found in the matrix of the mitochondria. So it means that if there is no mitochondria, there would be no gluconeogenesis for the pyruvate, unless for the glycerol. But mitochondrial matrix is very essential because it accommodates a pyruvate carboxylase enzymes. And of course, it is activated by acetyl-CoA. So at this point, we should understand that the pyruvate carboxylase 1 found in the mitochondria, and the enzymes require a coenzyme called biotin for it to work. And then the enzyme is also activated by acetyl-CoA. So acetyl-CoA is what facilitates and activates the key enzymes of gluconeogenesis. And then the next thing, so this is what actually happened in this reaction. First, you have pyruvate carboxylase, which is covalently attached with a biotin, and of course, this is the biotin here. So we have a biotin here, which is covalently bonded with the lysine residue of the amino acid, of lysine residue of the enzyme, which is carboxylase. sorry, which is the pyruvate carboxylase. So in the pyruvate carboxylase, there is an enzymes. Sorry, in the pyruvate carboxylase, in the active site of the enzyme, there is an amino acid called lysine, and this lysine is the amino acid that attach covalently with the biotin. And of course, look at what happens. So the carbon dioxide from the bicarbonate is activated and transferred by the pyruvate carboxylase to its biotin prostatic group. So it means that the CO2 from the bicarbonate, it will now be transferred to the biotin. And of course, this is the acetylco A plus here, indicate that it is an activator. It activates the enzymes. So with the utilization of the ATP after the biotin is actually being transferred with the CO2. So this is the CO2 that attach in the enzymes. So the CO2, after the CO2 is being added, then the enzymes will transfer the CO2 to pyruvate, thereby generating oxaloacetate. And this is the oxaloacetate. So the CO2 will be added here and oxaloacetate is generated, and finally the enzymes will now recycle itself. So then when this happened, what next? Of course, the oxaloacetate cannot cross the mitochondrial membrane and it is reduced to mallets. And of course, the mallet can be transported or can transfast the membrane. So that is what happened after you generate oxaloacetate. Oxaloacetate will now give us so the mallet. So after this mallet, because it can easily, the major reason why the oxaloacetate is converted into mallet is because the oxaloacetate cannot cross the mitochondrial membrane. So as a result of that, it must be converted to mallet using a mitochondrial mallet dehydrogenase enzymes. And then, of course, it will now transverse and transport it to the cytosol. And then the mallet will now be reoxidized, which is oxidatively decarboxylated to phosphoenol pyruvate by the cytosolic isozymes of phosphoenol pyruvate carboxykinase. So therefore, this is the mallet. So the cytosolic mallet dehydrogenase will now convert the mallet into oxaloacetate. And then the oxaloacetate will now be converted into phosphoenol pyruvate using an enzyme called phosphoenol pyruvate carboxykinase. So this is what happened in gluconeogenesis. In gluconeogenesis, you have the conversion of pyruvate to phosphoenol pyruvate must be in 2 steps and 2 enzymes. So this is where one of the other enzymes is added that make the gluconeogenesis to have 11 enzymes or 11 steps. So pyruvate carboxylase and then phosphoenol pyruvate carboxykinase. And of course, in the glycolysis, the enzyme is only pyruvate kinase. So then after this, first, this is the brief step of what happened in the mitochondria. Pyruvate will be converted to oxaloacetate, CO2 is utilized, and ATP. And of course, the enzymes require a coenzyme called biotin. And then the next thing after this, then the oxaloacetate will be converted into mallet, and then the mallet will now cross the mitochondrial membrane, and then in the cytosol, the mallet dehydrogenase enzyme, which is the cytosolic one, will now convert it into oxaloacetate. And then this oxaloacetate will now be converted back into phosphoenol pyruvate. And of course, a GTP is required in this step. And ADH plus is also generated. So in the step 1, we get CO2 from bicarbonate, and then the CO2 bind to prostatic group biotin, and then the enzymes for pyruvate carboxylase transfer the CO2 from the biotin to pyruvate forming oxaloacetate. Then in the step 2, oxaloacetate cannot cross the mitochondrial membrane, and then the enzymes malate dehydrogenase will reduce the oxaloacetate to malate, and then the malate will now leave the mitochondria to the cytosol, and then finally in the cytosol, the malate is reoxidized to oxaloacetate by cytosolic malate dehydrogenase, and then step 4, using a phosphoenol pyruvate carboxylase, the oxaloacetate will be decarboxylated to phosphoenol pyruvate, and then the oxaloacetate will gain a phosphate group to become phosphoenol pyruvate. So then the next thing is the regulation of the phosphoenol pyruvate pyruvate carboxylase, the action by acetyl-CoA. So the acetyl-CoA divert pyruvate, usually it divert the pyruvate from oxidation, that is the pyruvate dehydrogenase complex pathway, and pushes it towards the gluconeogenesis, that is pyruvate carboxylase. So how? The acetyl-CoA is a biomolecule that inhibits the pyruvate dehydrogenase complex. So it inhibits it and then stop or reduce the glycolysis and stimulate pyruvate carboxylase to start gluconeogenesis. So that is what happened. The acetyl-CoA, it inhibits pyruvate dehydrogenase complex and then activate pyruvate carboxylase. That is why the gluconeogenesis starts. Then the positive regulation. So high acetyl-CoA will stimulate the enzymes pyruvate carboxylase biotin to make more oxaloacetate. Then the oxaloacetate will proceed the pathway to produce more glucose. So the negative. So high level of acetyl-CoA inhibits pyruvate dehydrogenase complex and stop or reduce glycolysis. So the pyruvate dehydrogenase complex, the main function is convert pyruvate to acetyl-CoA. So once the acetyl-CoA are generated, then the acetyl-CoA will now go back to inhibit the pyruvate dehydrogenase complex to stop the citric acid cycle. So this is what happened. You have a pyruvate, and of course, in the presence of acetyl-CoA, the acetyl-CoA here will now inhibit the pyruvate dehydrogenase complex. And of course, at this stage, the fatty acids, of course, this acetyl-CoA, the acetyl-CoA is usually generated in two major pathways. It's usually generated in two major pathways. Number one, it's usually generated due to the oxidation, that is the beta oxidation of fatty acids. So it's usually generated through the beta oxidation of fatty acids. And then also by the action of pyruvate dehydrogenase enzymes complex. So oxaloacetate and oxidation of fatty acid and the action of pyruvate dehydrogenase complex are the two major pathways that generate a lot of acetyl-CoA. So once these acetyl-CoA are generated, then it will now inhibit the pyruvate dehydrogenase complex. So therefore, the citric acid cycle will be halt, will be stopped, will be blocked. And then at the same time, this high level of acetyl-CoA will now also activate pyruvate carboxylase, and then pyruvate will be converted to oxaloacetate, and then the gluconeogenesis will now be activated. So gluconogenic pathway will be activated in the presence of high amount or high concentration of acetyl-CoA. And then the next thing is the next step where we have fructose 1,6-bisphosphate. So this is a dephosphorylation pathway where the fructose 1,6-phosphate is dephosphorylated to fructose 6-phosphate. So at this stage, we also have activators and the inhibitors. So generally, a high concentration of AMP, that is adenosine monophosphate, and fructose 2-phosphate, when you have high level of these biomolecules, then AMP and fructose 2-6-bisphosphate will now inhibit the enzymes, which is fructose 1,6-bisphosphatase. So these biomolecules usually inhibit the fructose 1,6-bisphosphatase. Then also at the same time, the ATP, that is high amount of ATP, also it work actually in activating fructose 1,6-bisphosphate. So fructose 1,6-bisphosphate, it usually activates the fructose 1,6-bisphosphatase. So it means that these are the two opposing biomolecules that activate and inactivate, or that inhibit or stimulate the fructose 1,6-phosphatase activity. So the activator is ATP and the inhibitors are AMP and fructose 2,6-bisphosphatase. And of course, fructose 1,6-bisphosphatase is the enzymes that replace the fructose that replace phosphofructokinase 1. And then another enzymes is actually the dephosphorylation of glucose 6-phosphate, where glucose 6-phosphatase is an enzyme that remove the last phosphate and then generate glucose in the gluconeogenesis. So this is an enzyme that remove the phosphate group and finally generates glucose and the last step. And in addition, so this is the phosphorylation of glucose 6-phosphate. It allows the release of free glucose from the liver and kidney into the blood for the maintenance of the blood glucose level over the transport of other tissues for energy generation like brain. So then the next thing is the gluconeogenesis is an energy consumed process. So 6 energy phosphate bond from pyruvate to glucose are utilized. So at this stage, 4 ATP is is utilized, 2 GTP is utilized, and then 2 NADH are also used. But finally, when the glucose is obtained, then glycolysis will give us about a total of 38 ATP. So it is worth it. Also, we can recycle the non-carbohydrate precursors into the glucose. So it means that before you convert the oxaloacetate here into glucose, you need to utilize 2 ATP here, another 2 ATP here, so making a total of 4 ATP, 2 GTP, and 2 NADH. So it's an energy consumed pathway. It consumed energy. And of course, after the glucose is generated, about 38 ATP can be obtained from that glucose. So that is why it is worth it. So then the next thing is the regulation of gluconeogenesis. So the regulation of gluconeogenesis can take place in 3 different stages. There's a reciprocal control, allosteric control, and then hormonal control. So of course, in this reciprocal control, gluconeogenesis and glycolysis are the two opposing pathways. So if gluconeogenesis is taking place, then glycolysis will be stopped. And if glycolysis is stopped, then gluconeogenesis will continue. So this is a reciprocal pathway, is a two opposing pathway which are usually controlled by hormones. So we have allosteric. So of course we have acetyl-CoA. Of course, whenever you have an acetyl-CoA, so acetyl-CoA activate pyruvate carboxylase, and then also ATP also activates fructose 1,6-bisphosphatase, but AMP and fructose 2,6-bisphosphatase usually deactivate or inhibit the fructose 1,6-bisphosphatase. And then another one, we have glucagon insulin pathway. So when the concentration of glucagon is increased, it means that it stimulates gluconeogenesis pathway. But if the amount of insulin is increases, then it will now inhibit gluconeogenesis, thereby it will now activate glycolysis. And then there is also allosteric control. So in the case of allosteric control, the fructose 2-cis bisphosphonates, sorry, the fructose 2-cis biphosphates, they are actually the inhibitor, and of course the induction. We have phosphoenol pyruvate carboxykinase. So these are the three regulatory points of gluconeogenesis: glucagon and insulin cycle allosteric, and then reciprocal control. So these are the take-home message. So gluconeogenesis is an important pathway for glucose production from non-carbohydrate sources during prolonged fasting. Lactic glycerol and glutamate and gluconogenic amino acid are the major gluconogenic substrate. And then gluconeogenesis is not a simple reversal of glucose. In fact, gluconeogenesis requires 4 unique reactions to circumvent the 3 irreversible reactions of glycolysis. And then gluconeogenesis and glycolysis are reciprocal control, allowing efficient glucose metabolism. And it is mainly anabolic that consumes ATP for the synthesis of glucose. So these are the take-home message for gluconeogenesis. And these are some quiz questions. They are 10. These are some of the quiz questions, and I want you to attempt it and answer the questions on the comment section. Thank you very much. And if this is the first time you are coming to my YouTube channel, please subscribe.